Okay guys, in this particular lesson what we're going to cover is cellular diseases. Then we can move on to another type of cellular transport impairment. So before we were talking about basically osmosis, which is kind of like diffusion. We talked about channel transport. Now what I'm actually talking about is endocytosis, receptor mediated specifically. So this is a disorder that occurs due to improper endocytosis. FH, familial hypercholesterolemia. Hopefully we can break this word down with our knowledge of medical terminology. Hyper meaning excessive, cholesterol, pretty self-explanatory. Emia, we're talking about a condition of the blood. So this is basically a condition where there's too much cholesterol in the blood. And the reason for the excessive cholesterol in the blood is actually because of the LDL receptors. And I just want to, on a side note, talk about LDL versus HDL. LDL versus HDL, we have LDL is our low density lipoprotein, while HDL is our high density lipoprotein. And what I typically tell everybody to remember is that the LDL is the healthy, or I'm sorry, the HDL is the healthy, so H is for healthy, and this is really about the different types of cholesterol, and because the LDL, which is the bad type, because those receptors, there's too few or none at all, what actually happens is, as you can see in the picture, cholesterol is meant to attach onto this LDL, this particular type of protein, this carrier molecule, and it actually, on the receptor mediated, here's our receptor protein, it's actually going to help the cell dimple in and form that vesicle that's going to transport the cholesterol into the body. If there aren't enough receptors on the cell membrane to help transport the cholesterol into the cell, the cholesterol is not going to move into the cell where it's supposed to be broken down. If it doesn't get broken down, then it's never going to be sent to the liver to be excreted. If it's never excreted, then it's stuck in the body, and ultimately it's stuck in that space outside of the cell, which ends up actually being in the bloodstream. So that lack of receptors ends up creating excessive cholesterol in the blood, and that excessive cholesterol in the blood can lead to plaque buildup, and that plaque buildup can lead to heart disease. So this can be a pretty substantial, and we should recognize that the idea that familial is referring to the idea that it's hereditary, it's inherited. Then some of our cellular division diseases, and I think that the easiest thing for us to really think about, obviously, is going to be cancer. All right, that's the first thing that's obviously going to come to mind. Abnormal cell growth. What we're talking about is we're talking about cancer, or the potential for cancer. So neoplasia, right, is really talking about a neoplasm, which is just an abnormal growth, right? It's not a tumor yet. It's just an abnormal growth. Nothing cancerous necessarily at that point. It's not actually a cancer tumor at that point. It's just an abnormal growth. Something in the cellular division is occurring incorrectly, and because that cellular division is occurring incorrectly, you have extra growth or incorrect growth in that particular area. A neoplasm can then become a tumor, the tumor being an actual mass, not just recognizing abnormal growth, tumor being an actual mass. We should know benign tumors are non-cancerous, while malignant tumors are cancerous. But the interesting thing that people don't necessarily recognize about benign tumors is that, yeah, they're non-cancerous, but they're abnormal growth. It's a mass of cells, and that mass of cells is going to potentially continue to grow, and as it continues to grow, it's going to become larger, and as it becomes larger, it's going to interfere with that healthy surrounding tissue, and it can actually become a problem. So just because it's benign doesn't necessarily mean it's not a problem. It just means that it's non-cancerous. It could still potentially become a problem at later stages. Versus the malignant tumor, we know is cancerous, and that cancer cells are going to be invasive. Those cancer cells are going to continuously divide, and they're without a doubt going to extend into the surrounding tissue and start to affect the surrounding tissue, and they're going to continue to spread. We should also be familiar with the term metastasize. Sorry about that, guys. Metastasize. And what metastasize means is basically just spreading. And we should recognize the idea that once cancer cells are going to reach the bloodstream, they are going to metastasize. They are going to spread into different areas because the bloodstream is kind of a direct route for them to get everywhere in the body and spread to all those different areas. Then we can also, when we're talking about cell division disease, we can talk about oncogenes. And what oncogenes actually do is that they activate other genes that increase cell division. So realistically, the oncogene isn't actually what's triggering the cancer. I think we are familiar with the idea, the prefix onco implying cancer. Oncogenes aren't actually the ones that are causing it, but they're actually stimulating the genes that control the cell division. So what happens is that once they stimulate those genes then are going to secondarily increase the cell division. So it's kind of like a domino effect. Versus the next one, the tumor suppressor genes. Our tumor suppressor genes keep division in check. That's my check. All right, so it keeps division in check. Basically, it regulates cell division and makes sure that cell division is occurring at a proper rate in proper areas. But if there is a flaw, an error in those tumor suppressor genes, in those genes that are meant to kind of control the cell division and make sure it doesn't get out of control, if there's a mistake, then the tumor suppressor gene isn't going to properly keep cell division in check, and then cell division can get out of control. These are the two different genetic links, or kind of ways that we group genetic links to cancer. So this is just kind of a picture showing you about how cancer kind of can travel and move. So the cancer trigger being an inherited mutation or an environmental problem that creates the mutation, which we talked about in biology class, the idea that mutations can be something that you're born with, or they can be something that's caused due to an environmental factor. Like we know that skin cancer is directly linked to sun exposure, which is an environmental factor. And what actually happens is that because of that trigger, we either have an oncogene that gets turned on, and if the oncogene gets turned on, then the oncogene triggers a gene that increases division. Or this is a typo, the tumor suppressor gene gets turned off. And if the tumor suppressor gene gets turned off in the mutation, then the suppressor gene, the gene that's supposed to suppress or keep the division in check, isn't able to do its job, so cell division gets out of control. And the idea that those other mutations, we wind up with a loss of reproduction, with loss of specialization. It winds up traveling into bloodstreams. It winds up invading nearby cells. It ends up metastasizing, and it kind of creates a spiraling effect